A plasmid is a cellular structure formed by DNA that replicates independently from the chromosomal DNA. In bacteria, it provides the genetic information for certain cellular activities, like resistance to antibiotics. A typical plasmid is a double-stranded DNA molecule and circular. The size of the plasmid existing in nature varies from 1 to more than 1,000 kilobases. Many plasmids use the same replication machinery of chromosomal DNA and contains a replication origin, or ORI. Plasmids self-regulate its copy number through a repressor, a protein of RNA, which prevents many copies of the plasmid to be replicated. These repressors can also act on other related plasmids, thus excluding them from the cell. Generally, plasmids are classified into those which are in a low number of copies per cell, 1 to 10, and those present in a high copy number, 10 to 100. There are several methods to purify plasmids, but all of them follow the following basic steps: the lysis, the removal of large particles, the intermediate purification, the high resolution purification, and conservation, which consists of dissolving, freeze-dry, and fermentate. Plasmid DNA extraction is more complicated because it involves separating the plasmid from the genomic DNA. The lysis process must be quicker when dealing with plasmids to avoid irreversible denaturalization. Next, the sample is neutralized in potassium acetate solution to re-enrich the plasmid. Because plasmid is small, it can easily be annealed, but the genomic DNA is too long to re-anneal properly, and instead it becomes tangled so the complementary strands stay separated. The alkaline lysis method is used mostly for the isolating circular plasmids from bacterial cells, and it is based on the selective denaturation of chromosomal DNA by alkalization with sodium hydroxide and addition of SDS, a detergent, in conditions where the plasmid DNA remains close to its native structure, mainly due to its small size and circular supercoated nature. By neutralizing the medium and adding a high concentration of salt, in this case potassium acetate, precipitation of proteins largely due to treatment with SDS and the high salt concentration is reduced. It also precipitates chromosomal DNA, probably because reassociation occurs randomly among different regions of the DNA and thus insoluble aggregates are formed. By contrast, plasmid DNA does not precipitate, remaining in the supernatant. Now, lysozymes are hydraulic enzymes that damage bacterial cells walls by catalyzing hydrolysis of 1,4-beta linkages between N-acetylmuriatic acid and N-acetyl-D-glucamacin residues with a peptidoglycan and between N-acetyl-D-glucosamine residues in ketodextrins, and thus allows the extraction of the plasmid DNA. Plasmid can be visualized on agarose gels in 3 different forms: supercoiled, relaxed, and linear forms of full length.
The gel consists of 7 lanes. The first lane is the 1 kilobase per DNA ladder from oxygen. Lane 2 to 5 are the lanes from the other teams of Group 3 of the Laboratory of Genetic Engineering. Lane 6 is the sample of our team of the transformed E. coli with the XZFX gene from the plasmids of said transformants. In the gel, you can see 2 isoforms and 1 band from RNA contamination. Lane 6 is the lane from the team, and lane 1 is the 1 kilobase per DNA ladder from oxygen. The steps consist on acyl lysis with sodium hydroxide, SDS, and lysozyme. RNAs could be integrated. Then a neutralization with a high salt buffer that makes the precipitation of proteins and neutralizes the DNA, and a purification by centrifuge and phenol. It is very important to control the time so that only the plasmidic DNA can exit the cell and that the lysis creates just the pores necessary for the plasmidic DNA to exit. It is required a good integrity and purity, as there are no degradation signs in the gel.
The solution GTE or S-TET buffers and lysosome solution are used for the lysis of the cell, and ethanol is used to neutralize and precipitate the plasmidic DNA. In an agarose yield, the plasmid has to show two or three isoforms: relaxed, linear, and supercoiled. The first isoform in the yield should be the relaxed one, as its volume is higher than the other two. The second should be the linearized isoform, as it migrates easier than the relaxed isoform. The last, it should be the supercoiled isoform, and its volume is the tiniest and it migrates easier than any other through the agarose. In our results, we can observe only two isoforms, supercoiled and linear. The first is quite degraded, almost not visible, and located between 6,000 and 5,000 kilobases. The supercoiled isoform is located between 2,500 and 2,000 kilobases. This could be caused by an excessive degradation of the plasmid.